to be honest.
Fine, thank you. I think we move on to the next presentation on Seligo, a new class of customizable affinity reagents for diagnostics and therapeutics, presented by Dr. John R. Thornback, Managing Director of Apta Biosciences Ltd, Singapore.
Okay. Right, well, I'd like to thank the organisers for putting me back in Singapore. I wasn't due to go back until Sunday night, so it's quite nice to be in Singapore again. Didn't expect this. It's where I live, actually. And secondly, I'd like to thank Dr. Woodford for explaining the benefits of oligonucleotide-based affinity reagents. What we have is actually the next generation beyond Aptamus. That's what we believe, and I'll explain to you what I think we are. Okay, so a little bit about Apta Biosciences. We're a new company. We were established a couple of years ago, based on a lot of technology developed by Fujitsu. Why a Japanese software and hardware computer company should have a life sciences laboratory doing this work is a subject for a few beers. But anyway, what we do, we've got a novel technology, which will proprietary technology based on a lot of work developed by our Japanese colleagues. We're based in Singapore in the UK, and we have a small biotech. What is a Seligo? Well, a Seligo is basically, it's a proprietary affinity molecule. It's small. It's a DNA amino acid hybrid. And it's engineered such that what we have is that we have a peptide binding sequence, which makes us very different from an aptamer, which is actually a DNA binding sequence. We actually have a peptide binding sequence within a DNA backbone. Again, we have the advantages of fully synthetic, GMP manufactured, ISO 1345 certified manufacturing process. We will be making it. We can make display libraries, and we can make a whole variety of different display libraries. The one we typically use has 10 to the power 13 different molecules. Okay, well, I think we've heard this. Why antibodies have limitations? Okay. Want to do it. There are other technologies out there, and they all have their advantages as well. And I'm not going to go through that. But anyway, so what do we have? Well, our Seligo has a DNA scaffold, as I said, with amino acid side chains, which are the ones that bind to the target. And in case anybody asks me, actually, if you do take off the side chains, they don't bind. So I'm sure. Our basic Seligo structure in our current iteration is that we have a variable region which has 13 different amino acid side chains in it with a scaffold, DNA scaffold, and we have a site-specific attachment position. Now, we can, of course, alter that depending on, sorry, where's the thing gone? We can change the size of this region as we want, and actually, we don't actually need the scaffold. That has either. But what we've got is basically that we modify the DNA by putting the different amino acids on there, and we've got different properties that we have. We have 10 different amino acid side chains at the present time. And so what we have is that we've made 10 different amidites, essentially, which can be then put into a DNA synthesizer and make the Seligo. The Seligo structure, as we say, can be, in this case, with the variable region. You can imagine different indications. We start off, when we're doing the screening, with what's a 94, which has a variable region of 26, 13 different side chains, a double-stranded DNA stem, plus primers at either end. We can, of course, take those off if we want to make therapeutics, or leave one of them on as well. So basically what we have is a random libraries with these modified bases in different things. And we can, of course, make those using a simple DNA synthesizer. Actually, they're modified ones because you have to have 14 ports, of course, rather than 4 ports. So we have ACTG plus the 10 derivatives. But the core of our technology is that you can use our modified amidites on a standard DNA synthesizer, and we can also remove them all in one go as well when we want. This part of our analysis technology. So of course you kind of, as Dr. Wood just said, you can add things as well to these Seligos. What you can do is do either click chemistry or copper-free click, depending on what you want to do, and you can add, or do all the standard things like put fluorescent dyes and things like this on. And we've put radioisotopes on, we've put stable isotopes on, you can put drugs on, and make the molecules you want. So what's our process? Our process is actually, with aptamers, traditionally you've used the CellX process, and you go through multiple iterations. Actually, all we require is a single iteration to get at least nanomolar affinity. It's a very simple process. We have our screening. We have a screen. We take typically about 300 micrograms of the target material. To be honest, the basis of the technology is how good the protein is that we get. We do a negative selection. We can, if we wish to. We then screen against the library. We then knock off the DNA of the side chains. We then put that into an expression system, and we generate clones. And we harvest those clones. And then we characterize, very simple. Then we characterize. And typically it takes us around 7 weeks to get animals. So let me give you an example. So basically, the example I want to give you is dengue and West Nile virus. Now, dengue is a virus that's coming to you soon. I should tell you this. Dengue is a very interesting virus. There were 186 cases last week in Singapore. There's a very interesting post that says in Mexico last year, in the last few months, there's been 4,000 cases. Now, dengue normally is a virus which gives you a very bad headache and makes you feel sick for about a week. I had it about 18 months ago. But if you get it twice, there's a strong expectation you're going to die, because there were 4 different serotypes. And there were 992 cases in Mexico of the 4,000 had hemorrhagic fever. So where you have multiple variations of the virus, multiple serotypes, then you will likely to see hemorrhagic fever. Interestingly, the Sanofi virus does not work equally on all 4 serotypes. So our task was to develop Seligos against dengue, West Nile. They're 2 flavor viruses. Zika's the third one. Chikungunya is another one. Chikungunya is another virus that's going to come to us soon as well. So that was our task. Our collaborators, what we did was we picked the envelope protein, which is this one here. We took that virus, we took the envelope protein. There are 4 serotypes. West Nile virus is essentially the same. What we try to do is generate Seligos against all 4. I'll show you the one for the D2. And we went through the standard process, and we then selected 10 Seligos for both West Nile virus and in this case, and the dengue 2, I'll show you. We then developed an ELISA test, plus we started doing some interesting experiments to try and understand what the Seligos will do in other environments. This is typically what we get. From our data, we use BioCore typically as the first screen. From our clones, we generate 100 different Seligos. Remember, we have 10 to the power 13, so we select 100 hits from that. We take the best 10 of those. We then made those again, and you can see that in this case, in the West Nile virus, our Seligos actually do behave pretty well, as much as the best antibody that's out there as well. Interestingly enough, our West Nile virus one also detects a lot of different strains as well. So it's strain specific. Our dengue one, this is an interesting one. Can you detect different serotypes? Well, the answer is yes. This is our dengue, our dengue 2 specific molecules. They don't bind to dengue 1, dengue 3, or dengue 4, just a very small affinity to dengue 4. So we can detect serotypes. This is with a single screen, remember. And detect different serotypes. We can do, does it work in cells? Yes. Against cells, this is the plaque reduction exercise, PRNT test. It does inhibit the entry of dengue into the virus. So it's actually acting on, it says binding to cells as well as to individual proteins on the surface of the cell. They're stable in vitro, our molecules. We've had them structurally stable at room temperature and 37 degrees for 5 days. They're structurally stable in vitro in serum. and they don't impact on cell viability. So what we were looking for was we can develop, we can also, I haven't said, we can also develop molecules that bind to different isoforms of the same protein. We've done that with some protein biomarkers as well. So basically what we have is rapid development from the protein to the Seligo. We've got a single pass library screen, identify multiple candidates. We get high nanomolar affinity, low nanomolar affinity, high specific. They're sensitive, and they're relevant, and they're binding to specific proteins. And so we can modify them, they're structurally stable. And just to recap, the final bit is very much a similar one to some of the points that Dr. Woodward gave. They're quick to develop, high affinity, high specificity. They're small, robust, and stable. They're fully synthetic, easily functionalized, not limited to the immune system, no use for animals. They can be lyophilized. We do store them in lyophilized. We stored some for 2 years, lyophilized. And they can be a direct replacement for antibodies. This is where we fit, and we can argue about it later. So that's my final slide. Thank you very much. Bang on time.
Any questions?
Nobody else has any questions.
You mentioned Zika, among other things. What are the prospects of differentiating Zika from other
flaviviruses?
Like as dengue and...
Very easy, actually. We are in discussions with some partners in America to get a hold of the envelope proteins. We can differentiate between different serotypes of dengue. We can differentiate between those and West Nile virus, which is another flavivirus. We know we can differentiate chikungunya, which is another flavivirus as well. So the chances of having a Seligo that binds specifically to Zika is very high. For us, it's like all small biotechnology companies. You can only do a little bit, right?
So far as I know, I don't think any test is available yet.
There are quite a lot of PCR tests available, but there isn't an immunoassay test, as far as I know. Not a selective one yet. So yeah.
So, if there are no questions, I thank you for your presentation. We move on